for joining us for the 4th annual Black Immuno Week, a week we will be celebrating, amplifying, and supporting Black voices in immunology. Don't forget to tweet about our session today using hashtag ImmunoFlashTalk with each word capitalized for accessibility purposes. We will have live captions and ASL interpreters during the sessions. Please click on the CC at the bottom of the screen to access those captions. You can submit questions to the comment function on the platform, not the Q&A. And as a reminder, we require all audience members to be polite and respectful while in the Black Immuno space. If we see anything offensive or hear anything horrible, your comment will be deleted immediately and you will be removed from the platform. After the conclusion of this week, recorded videos we posted on our YouTube channel and transcripts will be made available on our website, blackandimmuno.org. My name is Kristen Carter, and I will be your host today to show some of our incredible Immuno Flash Talk finalists. At the end of the talks, please vote for your favorite Flash Talk video, and the Community Choice winner will win a prize. The link will be in the chat. We have 4 awesome Flash Talks today to share. We will start with the first one, titled, Now We Will Show, oh, I'm so sorry. Don't have the title just yet, but it is presented by Dr. Avolaji Olanjunju.
1979, it became obvious that since smallpox was eradicated in 1979, it became obvious that vaccines has been one of the most significant developments in contemporary medicine. And the administration of a dead or attenuated live vaccine induces an immune response towards certain pathogens and protects the host from a future challenge. Most effective vaccine formulations currently in clinical practice produce strong immune reactions and frequently require multiple inoculations to maintain a proper immune response for extended periods. In addition, the lack of comprehensive induction of cellular immunity by the available vaccine strategies has compromised their success against chronic infectious agents and cancer. An innovative and efficient method for developing a novel vaccine formulation is to use a live bacteria as a vector to deliver heterologous antigens. Live microorganisms are incredibly effective at inducing a long-lasting and targeted immune responses against specific antigens due to their immunostimulatory potential. Attenuated bacteria has been used to treat cancer for nearly a century, beginning from Coley's vaccine in the 19th century when you used bacteria to treat sarcoma, and fast forward to the use of BCG vaccines for the treatment of bladder cancer. And due to its ability to spread inside cells, Listeria monocytogenes is perfect for use as a live bacterial vaccine vector to boost the immune system's response to cancer and infectious disease. Hence, in this study, we'll be making use of the recombinant Listeria monocytogenes carryover bumigine, evaluating how the host with different genetic backgrounds controls the Listeria infection in terms of the innate immunity, and further, we will evaluate the ability to induce in vivo antigen-specific CD8 T cells and the ability to induce an effective anti-tumor immune response. And this study will contribute greatly to cancer immunotherapy and also immuno-oncology field. Thank you.
Thank you, Avalaji, for that wonderful talk on effective treatments for tumor therapy. I know it's a massive issue in the cancer field, and I look forward to future publications and seeing how you impact the community. Thank you. Next up, we will have Essie Ticella.
My name is Essie Chala from the University at Buffalo, and today I will tell you about sex-based differences in immune response to the pneumococcal conjugate vaccine. In general, there are sex-based differences in infections as well as vaccine responses, with male being more susceptible to infectious diseases, and they've also been shown to mount lower antibody responses to several vaccines compared to their female counterparts. So I wanted to investigate the role of biological sex in the context of Streptococcus pneumoniae. Streptococcus pneumoniae are bacteria that can cause very severe infections, including pneumonia and other invasive pneumococcal diseases. And again, males are more susceptible to these infections compared to females. There are vaccines on the market against these bacteria, including the pneumococcal conjugate vaccine, or PCV. In response to this vaccine, B cells produce antibodies dependently of T cells. And population surveillance has shown that following the introduction of PCV13, the incidence of invasive pneumococcal disease is still higher in males. So I wanted to know, what are the mechanisms behind these sex-based differences in the efficacy of PCV? For that, I turned to a mouse model where female and male mice were vaccinated with PCV13, and 4 weeks later, we infected them with the bacteria and monitored for survival. What you can see here is that about 90% of the vaccinated females were able to survive the infection, but only about 50% of the vaccinated males survived. So this shows us that male mice are less protected from PCV13. To investigate the mechanisms behind these differences, we looked at antibody levels in the blood of vaccinated male and female mice. Here we can see that for both IgM and IgG antibodies, males had lower levels compared to females. We next looked at antibody functions, and here I am just showing you a summary. The ability of antibodies to prevent the bacteria from binding lung epithelial cells was lower in males, but their ability to promote bacterial killing by phagocytes or by complement was unaffected. We then looked at T and B cell responses in the lymph nodes, and we found that upon vaccination, male mice had less of these T cells as well as less of the B cells compared to their female vaccinated counterparts. So in summary, male mice following vaccination with PCV13 mount a lower antibody response, leading to an overall lower protection. And with that, I'd like to thank my lab, especially my mentor, Dr. Buganan, and the Black in Immuno Committee for giving me the opportunity to present my research, and also funding sources. Thank you.
Thank you so much, Essie. I love the push for looking at sex differences in disease, and hopefully in the future, diseases will be able to be treated based off of identities, as well as those that are neither male nor female, and also those that undergo transformations. Thank you so much. Next talk will be by Iago Santos.
Thank you for stopping by. My name is Iago Santos, a PhD student at the Oklahoma State University, and my interest is elucidating the role of beta-defensin in stress response in fish. Stress is a very famous word nowadays, but not unique to humans. Stress represents a myriad of situations that can offer some risk to a living organism. For fish, stressor can be physical, like temperature, chemical, like hypoxia, and even perceived, like potential predators. I'm especially interested in the skin. The skin is the outermost line of defense against different dangers, offering not only physical but also functional protection. One interesting weapon used by the skin is called beta-defensins. Beta-defensins are small molecules, antimicrobial peptides that are highly conserved in vertebrates, what may reflect their crucial functions in the innate immunity. However, One question that has yet to be fully answered is how better defenses help the skin to protect the animal's body against stressful conditions. Using models of heat and transportation stress, rainbow trout is a powerful fish model that our lab leverages to understand how beta defenses may shape endocrine and immune response to stress conditions in vertebrates. Does beta defensin gene expression change in a stressor-dependent way in the fish skin? What is the impact of this change on the skin microbiome? How does stress impact the leukocyte modulation by beta defensins? And are beta defensins involved in the cutaneous stress response system in fish? Remember, it's always time to find your inner fish.
Thank you, Yago, for that awesome talk. As a fellow skin biologist, I firstly love the beat of the background music. Amazing. Second, love that you're studying the response of skin due to stress. Thank you, Yago. Next up will be Linda Akufo.
Hello, everyone, and you're welcome to my presentation. My name is Linda Ekufu, and I'm a research assistant at the Noguchi Memorial Institute for Medical Research in Ghana. I'm very passionate about research, specifically with viruses and immunotherapy. Okay, so I want to give a general background reason for this work. According to reports, there have been the emergence of possible drug resistance in the drugs that are currently being used for the treatment of chronic hepatitis B infections. Also, because of their long-term usage and then their side effects, there have been the need for the identification and production of new drugs that are safe, less expensive, and readily available for the use against the virus. This therefore brings me to the aim of this work, which sought to identify the abilities of Moringa and Philanthus plants to enhance the body's immune system to protect itself against chronic hepatitis B infections. Okay, first of all, I prepared these plants by cleaning and washing them, which was then followed by freeze drying to obtain fine powdered extracts. These extracts were then used to trigger cells collected from chronic hepatitis B patients to determine their ability to boost the immune system and also to prevent the spread of the virus and eventually to destroy them. From these graphs, I determined the plant's ability to boost the immune system by producing these cytokines, which are small molecules that aid the body's immune system in fighting against diseases, and in this case, hepatitis B virus. These particular cytokines have been found to prevent chronic hepatitis B infections. Therefore, the ability of these extracts to produce these cytokines indicates that the plants can help prevent chronic hepatitis B infections. In conclusion, I found out that these plants, or I will say that these plant extracts are non-toxic and they have the potential of preventing and treating HBV or chronic hepatitis B viral diseases. Thank you.
Thank you, Linda. That was an amazing talk. Love the idea of jumpstarting the immune system. It definitely brings back a question asked during Dimier's talk about what can we do to be more prepared for infectious diseases. Thank you all so much to all of our finalists. Don't forget to vote. The link is in the comments. Thank you to all the participants for recording the videos and participating in the competition. Please don't forget to vote for your favorite Flash Talk. We will announce the final winners next week on our Twitter. Thank you for joining this session. Please stick around for the next sessions. It's a one-on-one networking session, followed by a fireside chat with Dr. Corey Grayson at 2 p.m. Eastern Standard Time. And you can find all of us in the sessions tab when you go to the left and turn on your video and audio and participate. Thank you so much. Have a wonderful day.